Hi everybody. Today we're going to finish up Chapter 8 from the textbook. This is the Control of Gene Expression Part 2. Today we're going to start off talking about combinatorial control. This is a phenomenon where the cell can integrate multiple signals from both inside the cell and outside the cell and use those to make a type of molecular decision regarding whether or not it wants to express a gene. From there, we'll leap pretty naturally to coordinated gene expression in eukaryotic cells, and this is where different genes are turned on or off in a synchronized fashion to achieve a more robust and more whole cellular response. We'll talk a little bit more about this idea of epigenetics and how we can inherit gene regulation patterns, not the genes themselves, that's all classic Mendelian genetics, but inherit instead the regulation patterns of these genes so that new daughter cells, or in fact new offspring, are born with some type of molecular memory regarding which gene should be on and which gene should be off. And from there, we'll leap on to post-transcriptional control. We'll talk about microRNAs, very, very small RNAs that play a role in gene expression. The phenomenon of RNA interference, or RNAI, which is an incredibly interesting cellular process. And finally, long non-coding RNAs. And I should point out that this is our first instance in this course, I believe, where we're covering material that's exclusively found in the new 4th edition of our textbook. So these long non-coding RNAs are found only in the new edition of the textbook, and of course in this lecture as well. I just want you to be aware of that. So life in multicellular eukaryotic cells is very much like a symphony played by an orchestra. There are many, many different components to the cell that each add their own expertise, their own specialty to the grander whole vision of the cell in its totality. And what we mean by vision of the cell is, of course, just surviving. So different organelles play different roles in cellular survival, different proteins play different roles, and of course the genes that encode those proteins and the genes that see to the formation of those organelles also play different roles. So all of those processes and all of those players in the cell are coordinated. But what makes it interesting in terms of a cell is, unlike a symphony played by an orchestra, there is no central conductor in the cell. In other words, cells are highly synchronized, highly coordinated, insanely complex systems that have no centralized entity in charge, no single central coordinator for all of this. In order to achieve that, life in a cell is highly interdependent. What we mean is that every component of the cell keeps in close communication with, influences, and is influenced by every other pathway and process in the cell. And this is how the cell remains coordinated and in sync. In order to achieve this, it requires lots of different cellular processes, lots of different regulation schemes. Some of these you will have covered or will cover in the future in biochemistry. Those are the protein regulation schemes, the enzymatic activity regulation schemes, but also, of course, quite a bit of this comes from gene regulation itself. In addition to that, once a cell has gone down a given developmental pathway, when it is part of a larger multicellular organism, so let's say, for an example, once a cell is destined to become a liver cell, when that one cell divides into two, each new daughter cell can't start from scratch. Instead, each new daughter cell must be born knowing that it is to be a liver cell. It must be born as a liver cell. Said another way, it must be born already knowing how to be a liver cell. Said one third way, it must be born knowing which genes it needs to express to be a liver cell and which genes it should not be expressing if it's a liver cell. In other words, there's no time to re-differentiate from a brand new undefined cell to a fully differentiated liver cell with each round of cell division. In order to achieve this, we need some type of cellular memory. The new cell must maintain some type of memory from the previous mother cell regarding what identity it should have and what genes it should express. So in this lecture, we're going to talk about transient, short-term regulation as well as long-term static regulation where genes are permanently turned on or permanently kept off. So we're talking about the analogy of a light switch where we can flick back and forth between two processes as many times as we need to. That's the short-term transient regulation, as well as static regulation. In other words, if we want a gene off, we keep it off permanently for the life of the cell. This would be analogous to cutting the power cord to a certain appliance. Once that power cord is severed, that appliance is off and it's never being turned on again. That's our example for long-term static regulation. So eukaryotic genes are regulated by combinations of proteins. Even single genes can be regulated in this way. At the end of our last lecture, we said that unlike bacterial cells, eukaryotic transcription factors bind to these structures called enhancers. These are DNA sequences that lie thousands and thousands of base pairs away from their target gene. And in bacteria, regulators tend to bind very, very close to the promoter of the gene or operon that they're binding to. And enhancers in eukaryotic cells can, of course, be upstream of the target gene that's being regulated, but they can be downstream as well. What this means is that regulatory sequences for our genes can be dotted along very long stretches of the entire chromosome. And in between those kind of islands of enhancers, there can be unrelated DNA sequences between each one. There can even be other genes between those enhancers and the target gene. So what we wind up with is a situation such as this. Here we see a pink DNA region being bound by a red transcription factor, an orange region bound by an orange transcription factor, blue bound by blue, and yellow bound by yellow. Each of these DNA regions here is an enhancer. It's a DNA binding sequence specific for these transcription factors. And each of these colored shapes that you see bound to those regions are, of course, the transcription factors themselves. Each of these enhancers is specific for this gene that we're looking at right here. Here's the TATA box of this gene. Here's the gene itself, the coding region of the gene. And you can see that each of the transcription factors bound to its enhancer is targeted somehow, which we'll talk about in a second, to this gene. But all of the intervening DNA between one enhancer and the next is looping out, making room for those transcription factors to all come together. This becomes important because in eukaryotic cells, genes are turned on or turned off by committee decision. So each of these proteins that you see here, each of these transcription factors bound to an enhancer, has its own say or has its own vote regarding whether or not this gene should be turned on or off. Very few genes in eukaryotic cells are regulated by just one transcription factor. Very few genes in our cells are in two-state situations, in other words, on or off, in response to tryptophan being present or tryptophan being absent, much in the way that the TRP operon was regulated in the previous lecture. Instead, genes in our cells are turned on and off due to any number of different states that the cell might be experiencing at any time. In other words, if multiple proteins are needed to turn a single eukaryotic gene on or to turn it off, then what we have is the potential for genes to be responsive to multiple signals coming in through those multiple transcription factors. So genes in our cells can be responsive to time of development, to condition, what's around outside the cell, what's around inside the cell, to the amount of some substance, is there a lot of something, is there a little of something, etc. Each one of these transcription factors bound to its own enhancer transmits or carries with it a signal regarding some condition that the gene should be responsive to. And this is what we mean by combinatorial control. Combinatorial control is groups of regulatory proteins bound to their individual enhancers working together, voting together, to precisely control the expression of a single gene. In our cells, for example, a typical gene is controlled by dozens, if not hundreds, of different regulatory proteins, each lending their own signal to the overall regulation of that gene. Some of these regulatory proteins are activators, and so they're voting to turn the gene on. Other of these regulatory proteins are repressors, and so they, of course, are voting to turn the gene off. Again, each of these individual transcription factors is regulating a gene in its own independent way. So So you can think of each enhancer as a landing pad for the transcription factors, as we've talked about before. But you can think of each transcription factor as its own little mini signaling molecule, carrying with it a single message regarding something about the conditions of the cell. The regulatory protein can be carrying a signal based on the time of development, the amount of some factor, some stimulus that's being felt inside the cell, or some stimulus coming from outside the cell, etc. And so that message coming from that regulatory protein influences the expression of the gene. Again, we're talking about a democratic system for gene regulation. Each transcription factor has its own say, has its own vote, and that vote, if you will, is informed by whatever that regulatory protein was receptive to. What I mean by that is if we are not meant to express a particular protein after day 7 of embryonic development, then we're going to have a regulatory protein made from day 8 on that tries to keep that gene off. That is that regulatory protein's vote. It is voting to keep that gene turned off because that regulatory protein is transmitting a signal on time of development. Now, it's great to think of these proteins sitting around in a very calm, orderly fashion and each voting, but nothing could be further from the truth. As I've said in lab a number of times by now, probably, the cell is a mess. It's a big bag of mess, and so much more analogous to a stock market floor with people just screaming and shouting to get their votes heard, rather than this orderly process of sitting and raising one hand. But the end result is the same. Many different voices, many different votes, many different says, each responsive to a different stimulus. So the end result is that gene expression in a eukaryotic cell is a committee decision. And once each of these different regulatory proteins have had their say or made their vote, it must be decided in which direction the scale tips. Do all of the inputs fighting and voting for the gene to be turned on outweigh all of the signals and votes for the gene being repressed, or is it the other way around, and the signals for keeping the gene off are the stronger signals, overpowering those to keep the gene on? This type of regulation, this combinatorial control, controlling a gene through the combination of different signals, is complex, but it allows true fine-tuning of gene expression where genes can be responsive to a multitude of different signals coming into the cell. Truly, when a gene is on in our cells, it's on by democratic vote. So that brings us to the mediator complex, the all-important integrator of these signals. The question on this analogy becomes who counts the vote? It's great that all of these different proteins have their say, but who exactly are they talking to? When these dozens, if not hundreds, of transcription factors bind to their enhancers, those intervening DNA regions loop out in order to allow those transcription factors to reach their gene, what are they actually doing? Back to this figure. What is that big light purple blob that these transcription factors are physically associating with? In eukaryotes, activators, transcription factors that activate genes, function much more to grant RNA polymerase II permission to proceed with transcription and leave the promoter, rather than recruiting polymerase the way that we saw activators do in prokaryotes. In other words, activators don't really affect RNA polymerase binding to the promoter in eukaryotic cells. That mechanism for activation is pretty specific to bacteria. Instead, they aid in the initiation of transcription, getting the process started. This mediator complex is a huge protein complex that literally sits perched on the rear end of RNA polymerase II, kind of envelops it like a cape. And what this mediator does, this multi-subunit mediator made up of dozens of different proteins, this huge complex that drapes itself all over RNA polymerase II, what it does is it relays information. That's all mediator really is. It's a relay complex. All those transcription factors, those repressors and activators that are voting for the status of the gene, are relaying their signals to mediator. So it's mediator that hears all of these different signals from activators and repressors and passes an integrated signal onto RNA polymerase II. The current thinking in the field is that somehow mediator receives all of these signals affecting a single gene, the positive signals and the negative signals, the activating signals and the repressive signals. It receives all of those signals through the transcription factors themselves, and it integrates those signals. After weighing all of those votes and combining and integrating all of those different signals, Mediator, it is believed, sends a final single result, a summative signal. You can think of it as the tally of the vote onto RNA polymerase II. Mediator itself can simply activate RNA polymerase II, allowing transcription to proceed, or repress RNA polymerase II, halting transcription right there at the promoter. Mediator sends one signal on to RNA polymerase II, but the signal it sends is informed and decided by all of the different transcription factors that have sent their individual votes to Mediator. So we can get repressive signals sent to Mediator, which result in no transcription of the gene, or we can get activating signals sent to Mediator, which result in transcription proceeding down the gene. Now, pointing to the importance of the mediator complex in general, its factors, its subunits, are conserved throughout all eukaryotes. There is a central core of mediator complex proteins found in all eukaryotic cells from yeast all the way to us, and those individual proteins are highly conserved in all species. And then attached to this central core are additional subunits that tend to vary between species and even vary between cell types within a species, so this seems to kind of be a tailored, more specific form of mediator. Usually, mediator contains about 20 different individual proteins, and it's very, very large in size. Moving on now to the coordinated expression of multiple eukaryotic genes, I want to highlight for just a second the difference in what we're talking about. What we just talked about with mediator is multiple signals influencing the expression of one gene, multiple different inputs influencing whether a single gene will be on or off. What we're talking about now is a single signal, it's difficult for me to say, affecting multiple genes. So rather than multiple signals affecting one gene, now multiple genes being affected by a single signal. We talked about operons in bacterial cells. It's mentioned quite a bit in genetics, and we rehashed it in our previous lecture here. Operons essentially allow coordinated control of transcription in bacterial cells. Actually, there's no better way to do this, no more efficient way to do this. In bacterial cells, you have multiple genes needed for a single cellular process, such as the metabolism of lactose, the production of tryptophan, etc. You take all of those different genes and you put them under the control of just one single promoter, one single on-off switch. That way, when you turn that promoter on, you make all of those gene products simultaneously because you need them all at the same time. And when you turn that promoter off, you don't make any of them because you don't need any of them at that time. Again, nothing works better. This is true coordinated control of gene expression. All the genes you need are on, or all the genes you need are off, and that's how it should be. But eukaryotic cells throw a monkey wrench into that. See, there's complexity in eukaryotic cells that make operons unusable. In addition to that, we only have 20,000 genes, but we certainly have more than 20,000 processes going on in our cells. What that means is that single genes, the proteins that those genes encode, are needed for multiple different cellular processes. And so we can't have genes clustered into operons because we may need a single gene for one process, needed for another process as well. In other words, we may need a single gene of an operon on in one cell type and not another. And so we can't kind of group these genes together under the control of a single on-off switch. Because of that, eukaryotic cells require a different strategy to coordinate gene regulation. As an example, think of mitosis in general, not the specifics of it. How many proteins do you think the cell needs to undergo mitosis? Well, let's think about it. You need all of the proteins to initiate replication. You need all of the proteins required to dismantle the nuclear membrane. You need all of the proteins needed to align the sister chromatids at the center of the metaphasic plate. You need all of the proteins needed to pull those sister chromatids apart. You need all of the proteins to reestablish and recreate nuclear envelopes. You need all of the proteins for cytokinesis. That's a lot. Hundreds? Probably thousands, actually. And the cell needs all of these proteins at specific times it needs all of the proteins for replication first. It needs all of those proteins at the same time early on in mitosis. It needs all of the proteins for cytokinesis much later. But it needs all of those proteins for cytokinesis at the same time at the very end of mitosis. So obviously this requires tightly coordinated gene regulation. But how is this achieved in a cellular environment that lacks operons, and in addition to that is subject to these democratic rules of committee decisions? I don't know if you've had the chance yet to be on any committee in your life, but trust me, committees are not good at speed. Things are very, very slow when they're voted upon and when they are created by the input of many different minds, and the same is true of proteins. So we've got ourselves a problem. How do we turn genes on rapidly and coordinatedly when we're subject to this committee vote? The answer is that most genes have their regulations finely balanced on the edge of being repressed or activated. And so since they're finely balanced on that edge, usually only one additional transcription factor is needed to tip the scales one way or another. In other words, one more activator, talking to mediator, will usually tip the scale in the favor of activation, and one more repressor in favor of gene repression will usually tip the scale in favor of repression. So we're right on that razor's edge, balanced between yes, transcribe, and no, don't transcribe. Now, imagine many different genes, all scattered around the genome because they're not clustered together in operons, all needed for the same cellular process, and all finely balanced on that razor's edge between yes and no. And now imagine that each of those genes has an enhancer, a DNA binding site, specific for a transcription factor, and imagine that that transcription factor, that activator, just bound to that enhancer. When that one transcription factor is made by the cell, and it binds to all of those enhancers, and it's an activator, it's going to tip the scale in favor of activation for all of those different genes scattered around the genome, and it's going to do it together at the same time because it binds those enhancers at the same time. That transcription factor was made because a specific cellular process, such as cytokinesis, is needed. And so all of those genes have their balances tipped towards activation at the same time, together, for the same reason, because that single transcription factor was made. This is coordinated regulation in a eukaryotic cell. This is how it's achieved. The analogy I like to use for this is the Homeland Security Advisory System. Many of you were probably pretty young when 9/11 happened, but I remember 9/11 quite vividly. I was in grad school at the time, in my early 20s, and I remember that day very vividly. And you might not even know that this system exists anymore, but soon after 9/11, the Department of Homeland Security developed this advisory system. And we were kept at these states of alert as a society. There was low or green, guarded or blue, all the way up to severe or red. And you can see what each of those colors means in the black font there that defines each of those states. Now, how is this an analogy for coordinated regulation? Well, you see, what was really going on behind the scenes was that all of the police departments and all of the military units, they had very, very defined things that they needed to do for each of these different colors. So if we were at a high orange state of alert, police departments needed to do very specific things. National Guardsmen needed to do very specific things. The larger military needed to do very specific things. The government had specific tasks to do, etc. And if we went down to yellow, they could stop those orange specific things, but they had yellow things that they needed to do, etc. So what would happen is the government would issue an alert saying, we have gone from blue to yellow. And that's all the government would have to do, was issue that alert through the mass media. And all of the police stations across the country knew exactly what they needed to do, because they had been pre-programmed and predefined what needed to be done in a state of yellow. All the military units knew what to do. All of the National Guard's bases knew how to prepare. Everyone kicked into action in a very coordinated and choreographed way, simply due to receiving the signal that we were now at yellow alert in the country. The cell is very much built the same way. The genes that need to be turned on at each of the different levels or signals in the cell is pre-programmed into the regulation of the gene. All that needs to be done by the cell is send that one signal. We are now at a state of cytokinesis. That one signal being sent in the cell kicks all of these different proteins into action in all of these different ways in a very coordinated fashion to allow cytokinesis to occur. Now, of course, there's no color code in the cell, there's no mass media in the cell, there's no CNN, there's no radio. So how does the cell send a signal out that all different components of the cell can hear? It does it through the production of a single transcription factor. So a single transcription factor is made. It binds to enhancers all throughout the genome, and because of that, all of these different genes kick on in the same way at the same time in a coordinated fashion to achieve one predefined cellular process, cytokinesis, or one predefined societal process, protecting us in the event of a terrorist attack. So let's have an example. Let's have a real-world example of eukaryotic genes being regulated in a coordinated fashion. Let's see an example of a single signal regulating multiple genes at the same time. And let's use as our example yeast, since that's our organism for our lab, and it's a very simple, the simplest eukaryotic organism to understand. And let's use a very simple signal, such as the presence or absence of oxygen. Whether we know it or not, we're all familiar with the fact that yeast can use molecular oxygen for respiration. If you've ever consumed bread, or even more informatively, if you've ever made bread, you know that yeast breathes. You add yeast, sugar, warm water, and flour. You mix those together, and you wait for that bread dough to rise. What's causing that bread dough to rise is carbon dioxide gas. Where that carbon dioxide gas is coming from is the yeast. You see, the yeast is taking in the sugar that you've added as well as oxygen from the air. And the yeast is breathing, very much the way we do. They are respiring at the molecular level. And the waste product of that respiration is carbon dioxide, same as for us. And that carbon dioxide causes the bread dough to rise. It gets trapped in little bubbles in the dough and causes the bread to proof. But we are also well aware that yeast can survive without oxygen. Many of us have enjoyed the benefits of yeast fermentation. If yeast is deprived of oxygen, it can still survive, it can still make ATP and energy, but it does it through the process of fermentation or anaerobic respiration, and the byproduct of this is not carbon dioxide, but instead alcohol. So yeast can survive in the presence of oxygen, and yeast can survive in the absence of oxygen. But I don't think it should come as a surprise to you that the proteins that yeast needs in order to respire are different than the proteins and enzymes that yeast needs to ferment. What that really means to us in a molecular biology course is that the genes that need to be turned on in the presence of oxygen are different than the genes that need to be activated in the absence of oxygen. So there's a whole set of genes for a whole set of proteins that allows yeast to respire, and there's a whole different set of genes for a whole different set of proteins that allows yeast to ferment. How do we achieve that? Well, in the nucleus of yeast cells, there are many, many genes, about 6,000 of them, but one of those genes is called ROX1. And just to give this a little bit of context, that ROX means something. It stands for regulated by oxygen, so we're already on the right path here. So the ROX1 gene is a gene that's regulated by oxygen. And there's a protein, a transcription factor, in fact, that binds to an enhancer or a DNA regulatory region upstream of ROX1, and that's the HAP1 protein. So the HAP1 protein is a transcription factor that binds to the ROX1 gene. And when oxygen levels are abundant and oxygen is available to the cell makes heme using that oxygen. That heme molecule cannot be made in the absence of oxygen. When there's no oxygen, there's no heme. And when there's no heme, there is no expression of the ROX1 gene. The ROX1 gene is repressed. So when oxygen levels are low, ROX1 cannot be made by the cell. But when oxygen levels climb and the cell is able to make heme, that heme molecule directly binds to and interacts with the HAP1 protein. This combination of heme and HAP1 together is a transcriptional activator, and it turns on the expression of the ROX1 gene. The ROX1 protein is made now as a direct result of oxygen being available in the cell. What is ROX1 protein? It itself is a DNA binding protein. It binds to the upstream region of all of the genes that are needed for fermentation. ROX1 recruits another protein called TUP1, and TUP1 is a very robust, well-accomplished transcriptional repressor. Now, all of the hypoxic genes are being regulated in this way. All of the genes that are necessary for fermentation are regulated in this way. So take a moment to reflect on this. When oxygen levels are high, yeast wants to express its respiration genes and repress its fermentation genes. When there's oxygen around, yeast wants to breathe. When yeast wants to breathe, it doesn't want to ferment. So what happens when oxygen is around? When oxygen is around, heme is made. Heme interacts with HAP1, and together they promote the expression of the ROX1 gene. That means we make the ROX1 protein. The ROX1 protein then goes and binds to upstream regions of all of the fermentation genes. It recruits this TUP1 repressor, and TUP1 turns all of those genes off. So all of the fermentation genes are off due to TUP1, which was recruited by ROX1, which is on because there's oxygen around. Now oxygen levels fall. Yeast has a decision: suffocate and die, or express the fermentation genes, ferment, and continue to survive in the absence of oxygen. But this all happens organically. This all happens naturally now, because as oxygen levels fall, heme can no longer be made. If heme can no longer be made, HAP1 is no longer an activator. ROX1 protein is no longer made. If ROX1 protein is no longer made, there's nothing around to recruit TUP1 to the fermentation genes, so TUP1 floats away. In the absence of the TUP1 repressor, we get expression of the fermentation genes, and so yeast can now ferment and survive in the absence of oxygen. As oxygen levels climb, heme is made again. Heme, along with HAP1, turn ROX1 on. ROX1 protein is made, binds to the fermentation genes, recruits TUP1, turns those fermentation genes off again. This is coordinated control. A beautiful, simple, and elegant system to turn multiple genes on or off together as needed due to a single signal in the cell. But why do it this way? Well, it's done this way for a reason. TUP1 represses lots of genes. Yeah, it represses all of the fermentation genes, that's true, but it represses lots of other genes as well. It represses genes that are encoding proteins involved in all different types of adaptation to environmental stressors, not just hypoxia. And TUP1 is a very, very robust, widely used transcription factor in yeast. In fact, it's referred to as the general repressor because no other single protein represses more genes than TUP1. So, some examples of this. The hypoxic or fermentation genes that we've just discussed are repressed by TUP1. We just said that. But so too are genes that are repressed in the absence or presence of glucose. Genes that are responsible for repairing DNA damage are repressed by TUP1 when there's no DNA damage around. All of these are genes that are needed in a hurry when they're needed. All of these are genes that need to be expressed when needed or the cell will die. And all of them are repressed by TUP1. But, of course, we don't want all of these different genes regulated together. That would be wasteful. In other words, we want the fermentation genes to come on when oxygen levels fall, the way we've just discussed. But we don't want glucose starvation genes to be turned on under that condition. We don't want the DNA repair genes to be turned on when we're starving for oxygen. That makes no sense. And of course, if the DNA is damaged, we want to turn on the DNA repair genes. But we don't want to turn on the fermentation genes because DNA is damaged. That would make no sense either. So all of these different classes of genes are regulated by TUP1, yet all of these different classes of genes are regulated by different environmental signals. how do you regulate different genes in different ways with the same exact repressor? Well, you do it by having different DNA binding proteins recruit TUP1 to the DNA. ROX1 recruits TUP1 to the fermentation genes. MIG1 recruits TUP1 to the glucose-starved genes. RFX1 recruits TUP1 to the DNA damage genes. So if you want to express the fermentation genes, stop making ROX1. TUP1 can no longer be recruited to the fermentation genes, and those genes will be turned on. But you're still making MIG1, so TUP1 is still repressing the glucose genes. You're still making RFX1, so TUP1 is still repressing the DNA damage genes. Your DNA is damaged? You need those genes on? Stop making RFX1. TUP1 recruitment will leave the DNA damage genes and they will turn on. But you're still making MIG1, so the glucose genes are still off, and you're still making ROX1, so the fermentation genes are still off. So you can achieve individual control of classes of genes using the same repressor protein based on which of these DNA binding proteins you make or don't make. ROX1, MIG1, RFX1. In addition to all of this, combinatorial control can also create different cell types within a larger multicellular organism. All of us, all human beings, went from a single fertilized cell to a fully surviving human being in about 35 weeks, a little bit short of 9 months. It's truly and utterly astounding. Go from one cell to an 8, 9, or 10-pound baby in such a short time. Distilled down to its most basic and molecular framework, what made this rapid development possible was nothing more than gene regulation, or said another way, nothing more than the synthesis of proteins. Different proteins made at exactly the right time and exactly the right way to make this possible. It's proteins that caused those embryonic cells to divide as rapidly as they did. It's proteins that caused those embryonic cells to take on a cell fate and develop into the specific cell types they were needed to be. It was proteins that gave these destined cells their specific functions. We'll avoid all of the detailed literature that's presented in the textbook for this section, but we'll keep the big picture in mind. It's still important to note that a single transcription factor can not only dictate whether a gene is on or off, but can solely dictate the fate of a single cell. But just like combinatorial control, just like coordinated gene regulation, even though a single transcription factor dictates cell's fate, it doesn't do it alone. It simply tips the scales. How again can we have a single factor tip the scales, yet not be working alone? Well, very much like what we just said for the gene regulation story, what this is based on is the set of proteins already made by that given cell. A single and final transcription factor is the last piece needed to seal the cell's fate and cause it to differentiate down its pathway. Here's an example. We have some precursor cell at the top. This is a totipotent cell. It has the potential to be any cell type in the human body. That cell divides into two. One of those daughter cells makes a specific regulatory protein. Let's say it's protein number 1. The other cell doesn't make that protein. Those cells divide. One of the daughter cells of the first makes regulatory protein 2. So too does one of the daughter proteins of the second cell. So now we have 4 different cell types or subtypes. We have one cell that has no regulatory proteins made, one that's only making number 2, one that's only making number 1, and one that is making 1 and 2. This is predisposing these cells to be different cell fates. When we talk about cellular differentiation, what do we really mean? What word is hidden in differentiation? Different. These 4 cells are different from one another. Therefore, they can become different things. As these cells divide, some of them make regulatory protein number 3 and some of them don't. And we have all possible combinations represented. No regulatory proteins, just 3, just 2, just 1. 2 and 3 together, 1 and 3 together, 1 and 2 together, or all 3 expressed. We now have 8 different cell types represented. Now, what this particular cell here on the end, this cell on the right-hand side, becomes, if it becomes a muscle cell, it's becoming a muscle cell solely because it is making regulatory protein 1, 2, and 3. Perhaps this cell here is becoming a neuron. It's becoming a neuron solely because it has regulatory proteins 1 and 2 only. This cell is becoming a lung cell because it is expressing 1 and 3 only. So let's come back to our muscle cell. Why is this a muscle cell and not a neuron? Why is the fate of this cell to be a muscle cell, a myoblast, and not a neuron? Well, it's because of the presence of regulatory protein number 3. See, neurons become neurons because they express 1 and 2. Well, this cell is expressing 1 and 2. Why is it not becoming a neuron? Because it's expressing 3 also. So the expression of protein 3 is what sealed the fate of this cell. If it was not for the expression of protein 3, this cell would have become a neuron. So did the expression of protein 3 seal the fate, be the deciding factor for the fate of this cell? Yes. Did it decide the fate of this cell alone? No, it didn't. Cells that express number 3 and only number 3 become some other cell type. This is going to be a muscle cell because it's expressing 1, 2, and 3, but the expression of protein 3 is what sealed the fate of the cell. The analogy here could be the UPS trucks that are so busy around Christmas time. See, each box that gets loaded onto the UPS truck determines the route that truck is going to take that day individually. First, the first box gets loaded, and that tells the truck what neighborhood it's going to go into. Then the second box gets loaded. And now we begin to piece a route together. Maybe we go to house number 1 first, and then house number 2. Then the third box gets loaded. Then the fourth box gets loaded. By the time the truck is full, the entire route is established for that truck that day. The last box to get put on the truck is what sealed the fate of the entire route. However, that last box did not dictate the entire route on its own. The rest of the route was determined by all of the other boxes loaded onto the truck before it. So it's the combination of all of the boxes that make up the route, with simply the last box defining the route. It's the combination of all of the regulatory proteins made by the cell that defines the cell fate, but it's that last final regulatory protein that seals the fate as to what the cell is going to be. So now let's get on to this idea of epigenetic inheritance, this molecular memory. How do we inherit gene regulation patterns in addition to the genes themselves? Again, as we said in the intro to this lecture, it would be very inefficient for a cell to have to relearn and reacquire its differentiation with each round of cell division. It would be inefficient for a liver cell to have to relearn to be a liver cell. There must be some kind of cellular memory so that the daughter cells of a cell of one cell type retain that cell type and don't have to redifferentiate. What we're really saying here, then, is that there needs to be a way for a cell to inherit regulatory patterns for its genes, because it's the expression of genes, it's the proteins made by cells, that give it its identity, that give it its cell type. This is accomplished, the cellular memory, in part by the histone code that we talked about very early on in our semester. Also, some proteins participate in what are referred to as positive feedback loops, where the proteins activate their own expression. In positive feedback loops, the protein is an activator for its own gene. So what happens is the daughter cell inherits some of the pre-existing protein, and that protein goes ahead and binds to its own promoter and drives expression of itself, making more of itself. So for positive feedback loops to work, the daughter cell needs only to inherit a small number of the pre-existing proteins made by the mother cell, and those proteins will see to the production of many more of themselves because they drive their own expression. In addition to positive feedback loops in the histone code, DNA methylation also allows for cellular memory. In our cells, cytosine can be methylated, and the methylation of cytosine simply serves as a mark or a flag. You might remember we talked about methylated cytosine in the context of DNA repair for bacterial cells. It's how, during DNA mismatch repair, bacterial cells could tell the old strand from the new strand. We have the same methylation mark in our cells, methylation of cytosine, but it's not to differentiate new strands from old strands. It's more for this epigenetic mark that we're discussing here. See, when eukaryotic genes are methylated, they are completely silenced. They are repressed long-term. The power cord to that gene is severed. Methylation patterns are heritable. Hopefully you can see the implication here. See, if we have a methylated gene in a maternal cell, that gene is off. It is completely silenced. Now, as that DNA is replicated for the process of cell division, we have two new molecules of DNA that are hemimethylated, very much as we saw in the DNA repair story. The old strand is methylated and the new strand is not. This is not the purpose of the methylation, but the consequence of replication is the same. We reestablish the methylation patterns in those new daughter cells by methylating the new strand so it matches the old, but remember what the methylated state promoted. Silencing of the gene. So now this gene is silenced in the daughter cells as well. We have inherited the gene regulation. The gene was repressed in the mother cell, and it is inherited in a repressed state in the daughter cell because we inherit these methylation marks and we maintain them in the daughter cell. I can't describe to you how stunning this was to the molecular biology community when it was first described. It was the prevailing wisdom for decades in molecular biology that the DNA sequence of genes were inherited, but that regulation patterns had to be relearned and reacquired. If a gene was repressed in the maternal cell, it had to be re-repressed all over again in the daughter cells. Not the case. At least when it comes to gene silencing due to DNA methylation, we can inherit the gene silencing patterns, we can inherit the repression. So that's almost 2 full lectures on regulating gene expression at the level of transcription. Everything we've discussed in this lecture so far and in the last lecture has been regulating transcription before it happens. Regulating the expression of genes at the first point of control along gene expression, transcription initiation. This is the most efficient way to regulate genes. It's the most energy saving. We can stop genes from being expressed before we even start and save all of that energy that it takes to make all of those messenger RNAs. However, we do need redundancies and we do need other forms of regulation. In particular, cells need a form of regulation. In other words, we started expressing this gene and it turns out we don't need it. We need to undo that transcription. We need to regulate gene expression after transcription has occurred so that we don't make a protein we don't need. Is it less efficient? Yes, it is. Does it waste the energy of making the messenger RNA? Sure it does. Yes, this is not the best form of regulation, but it's still good to have a failsafe and have a cellular mechanism to prevent the production of a protein that's not needed after transcription was accidentally allowed. In addition to the strategies we're about to discuss for post-transcriptional control, please keep in mind that alternative splicing, which we've already described, is also its own form of post-transcriptional control. But there are mechanisms for post-transcriptional control where even after the protein is made, it can be regulated. I'm sorry, those are post-translational controls, and those, of course, are more reserved for a biochemistry course that you have either taken already or will take after this course, and we'll leave those concepts for there. Biochemistry is essentially just a course in protein biology, whereas molecular biology is really just a course in DNA biology. Don't want to step on any biochemist's toes. So let's talk about nucleic acid-based post-transcriptional control. And more specifically, let's talk about the 5' untranslated regions of messenger RNAs and how we can use those to control the expression of a protein. The 5' UTR, or the 5' untranslated region, this is the very beginning of the messenger RNA that does not code for protein. Those regions of bacterial messenger RNAs often form secondary structures. They base pair with themselves, so some stretches of that RNA molecule base pair with other stretches, causing what we call intramolecular base pairing. We would refer to this as a stem loop. These pink rungs that you see here represent base pairs between one stretch of this messenger RNA and another stretch of the same molecule. This base pairing causes the region that is base paired to kind of become its own little short stretch of double-stranded RNA. And then this up here we refer to as the loop. This is a region of the RNA that is not base pairing, and so it just kind of loops out and gets out of the way. So we call this structure a stem loop. The stem is the base paired region, the loop is not base paired, and it forms as a consequence of one region of the RNA being complementary in base pair to another region. When we have this type of intramolecular base pairing in the 5' UTR of a bacterial messenger RNA, we can see the Shine-Delgarno sequence, which is also important for ribosome recruitment, being base paired with other messenger RNA sequences. That means it's unavailable for base pairing with the ribosomal RNAs of the ribosome, and that therefore interferes with translation initiation. You can't recruit the small subunit of the ribosome, you can't initiate translation, and of course then protein will not be made. Some signal or trigger must occur that disrupts these stem loops, these intramolecular base pairs. Once they're disrupted, they're no longer base paired. The Shine-Delgarno sequence here in orange is then free. It can interact with the ribosomal RNA of the small subunit of the ribosome, and we can get initiation of translation. And so we can get post-transcriptional control using these stem loop structures that sequester away the Shine-Delgarno sequence. Now, obviously, this is specific for bacteria because we don't have Shine-Delgarno sequences, but it's our simplest example for post-transcriptional control. We're making protein or not making protein at the level after RNA production, after transcription. We also have microRNAs. MicroRNAs are what they sound like, very, very small RNA molecules. They're very, very common in eukaryotic cells. We find them in worms, insects, mammals, plants, etc. MicroRNAs block the translation of a messenger RNA, but they don't destroy it. Instead, microRNAs bind directly to messenger RNAs, usually in the 3' UTR, or the 3' untranslated region, the region of the messenger RNA that comes after the stop codon. MicroRNAs have also been shown to base pair to the coding regions of genes. It's just not their common way of regulation. MicroRNAs are made on purpose by the cell. This isn't some kind of weird artifact. They are transcribed intentionally by RNA polymerase I and RNA polymerase III. There are chromosomal genes encoding these microRNAs. They are small. They're only about 70 bases in length. And they usually don't base pair perfectly with their target RNA. Instead, they usually have 1 to 3 bases that do not pair, and this results in what we refer to as a bulge. And so the microRNA here is in red. The target messenger RNA is in blue. We have a great deal of complementarity, but there are a few regions, 1 to 3 bases in each molecule that don't pair up well, and so we get this little bubble, this little single-stranded bubble there. Why is that bubble there? We have no idea, but what we do know is if we fix that bubble and we make a modified microRNA where the base pairing is complete, they don't work nearly as well to inhibit translation. So although we don't understand what this unbase paired region is doing, we know that it's important for microRNAs to inhibit translation as well as they do. When these microRNAs do bind to their target messenger RNAs, they somehow block translation by the ribosome. How do they do that? We don't really understand that either. We just know that they do. They may be interfering with translational initiation, but again, keep in mind we see microRNAs that bind to all different regions of the messenger RNA, and regardless of what region they bind to, they do inhibit translation. So that seems to suggest that it's not just as simple as getting in the ribosome's way. What is clear, though, is that microRNAs are used by some of the same cellular proteins to inhibit translation as play a role in another process called RNA interference. RNA interference is much more understood than microRNAs, and it is simply an amazing story. RNAI, or RNA interference, is a mechanism used by cells for gene silencing, for turning off the expression of genes in a very dramatic and robust way. RNA interference relies on the formation of double-stranded RNA molecules. Once double-stranded RNA molecules form and are recognized by the proteins that oversee RNA interference, all double-stranded and all single-stranded RNAs that have a sequence matching that initial double-stranded molecule will be destroyed. Period, end of story. In other words, if a double-stranded RNA molecule is observed in the cell, and it has the sequence in that double-stranded molecule that matches the sequence of a gene, when that gene is transcribed and that messenger RNA is made, even if it's single-stranded, it will be recognized, the messenger RNA will be recognized by the RNAI response, and it will be destroyed on site. And so you'll never be able to make a protein from that gene again, because every time that gene is transcribed, that messenger RNA will be destroyed, due solely to the initial presence of double-stranded RNA in the cell that matched that gene sequence. Any messenger RNA transcribed from that gene will be immediately marked for immediate destruction. End of story. RNAI might very well be the most potent gene silencing response. It's believed that the genesis of this came from a very primitive, evolutionarily evolved immune system response to defend us against viral infection. Many viral infections, and many of the worst viral infections, have double-stranded RNA as the viral genome. So by having this very robust mechanism for destroying double-stranded RNA and destroying all RNA that resembled that double-stranded RNA, we believe that perhaps this is a way that the cell evolved to fight viral infection. So how does it work? How does RNA interference work? Well, RNA interference is triggered by double-stranded RNA, as we just said, and it has to be of a certain length. It has to be double-stranded RNA that is fully base paired with no bulges, and it's about 21 to 23 base pairs of fully base paired region. If we have longer double-stranded RNA molecules that are fully base paired with no bulges, they are processed by the cell and cut into smaller fragments that are between 21 and 23 base pairs in length. And the enzyme that does this processing is called dicer. It's called dicer because it literally dices up these double-stranded RNA molecules. Dicer is a cellular protein. We all make dicer. You and I have dicer floating around in our cells right now. It's endogenous to our genome. These small, processed 21 to 23 base paired fragments of double-stranded RNA are called small interfering RNAs, or siRNAs, and they are loaded onto a second protein called RISC, which stands for RNA-induced silencing complex. And it's easy to figure out what RISC does. It silences genes, and it does so in an RNA-induced fashion. What RISC does is it takes that double-stranded 21 to 23 base pair length RNA molecule, and it separates the two strands. So it pulls the strands apart, and it uses one of those two strands from the small interfering RNA to sniff around, scout out, and recognize any other RNA molecule that matches or complementary base pairs to that small interfering RNA. Quite literally, the siRNA loaded onto RISC is giving the RISC complex the scent of all of the other RNAs to destroy. Many of you have probably seen on some crime show or another where you have a bloodhound and the cops are trying to find some criminal or some kidnapped kid or some person, and so they take an article of clothes from the person they're looking for and they give it to the bloodhound. They say, smell this, get this scent, get this scent. And then the dog goes on a sniffing rampage trying to find that scent somewhere. Give the dog the scent, and then the dog goes and finds anything else that smells the same way, including the person you're looking for. The RISC complex is the dog, and the small interfering RNA is the scent. By giving the RISC complex that small interfering RNA, you're saying, here boy, smell this, and any other RNA molecule in the cell that smells like this, that has this sequence, that base pairs to this strand, you destroy on site. So once the RISC complex recognizes any other RNA molecule that can base pair to the small interfering RNA it's carrying, RISC destroys that RNA molecule immediately. RNA interference is found in most eukaryotic cells, but it is uniquely eukaryotic. It's not found in prokaryotic cells at all. Again, it's probably the most potent form of gene silencing of any protein regulation known so far. Less than 50 small interfering RNA molecules are enough to silence thousands of pre-existing, already transcribed messenger RNAs. This is due to amplification, and the reason for this robust amplification of response is due to a third protein called RNA-dependent RNA polymerase, RDRP. What RNA-dependent RNA polymerase can do is copy small interfering RNAs so that a few, like 50 small interfering RNAs, can be made into thousands of small interfering RNAs. Where RDRP comes in is after RISC cuts an mRNA down to size, two unstable fragments are left. So now we have a cleavage of the target messenger RNA by RISC. The messenger RNA has been destroyed. We have two unstable fragments, the two halves of the messenger RNA that RISC just cut and destroyed. RDRP binds to one of those fragments and uses it as a template to generate a second complementary strand of RNA. See, like all RNA polymerases, RDRP does not need a primer, so it can bind to single-stranded RNA and just start making new RNA in kind. Once RDRP has made that complementary strand, what do we have? Well, we have double-stranded RNA, don't we? And double-stranded RNA is recognized by the dicer complex and cut into pieces. And then those pieces are given to RISC. And RISC will take one of the strands from those pieces and use it to scout out and sniff out other RNAs to destroy. Once those RISC complexes cut those RNA molecules, RDRP will bind to those single strands and make new copies. Those double-stranded RNAs will be substrate or fodder for dicer to cut them, and so the process continues to reiterate again and again and again and again. The double-stranded RNA is used by dicer, which generates more small interfering RNAs for RISC. RISC goes and cuts more target messenger RNAs, which are then amplified using RDRP. That's the amplification that we see. It's summarized, I think, very nicely in this figure here. Again, we have our long double-stranded RNA. It's cut initially by dicer to process it. The small fragments are taken up by RISC. RISC uses one of those strands to home in on other strands and cut those strands for degradation. Here's the RISC complex with one strand targeting it to the green messenger RNA, and RISC destroys that messenger RNA by cutting it. But the cut fragments can be amplified by RDRP. RDRP will make a second strand of RNA using a first strand as template. That new double-stranded molecule will be seen by dicer as template for processing. Once processed, those new molecules will be fed to new RISC complexes, which will use those single strands to home in on new target RNAs and therefore amplify the response. Mammalian cells cannot amplify RNAI. We can do the initial rounds of RNAI, but we lack the amplification aspect of it, and the reason why we lack amplification is because mammalian cells do not have RDRP. So without RNA-dependent RNA polymerase, we can't amplify our response, but we certainly do have RNA interference. Not surprisingly, if it occurs in nature, and it is of any use to a research laboratory and a research scientist, we will exploit natural phenomena for use in our laboratory, and RNAI is no exception to this rule. RNAI has widely been used in the laboratory setting to control and prevent and silence protein expression of targeted genes. Essentially, what RNAI offers the researcher is shutdown on demand. By intentionally expressing short interfering RNA molecules, scientists can achieve almost instant stoppage of protein synthesis in a very specific way. You can shut down the synthesis of a single protein in a very fast and robust way using RNA interference. Pretty powerful stuff. You can let a cell develop or function for days or weeks on its own natural program, and then hit it with a signal, cause the production of an siRNA, and deprive that cell of one single protein that you think is important, and then watch what happens. In the laboratory application of RNAI, artificial genes are made that, when transcribed, form double-stranded RNA in different ways, and then these double-stranded RNA molecules are processed by dicer and initiate the RNAI response. We can make double-stranded RNA by expressing a large stem loop that folds in on itself, and so the double-stranded RNA region is the stem. We can express one region of DNA from two different promoters, so we make one strand in one direction, the other strand in another, and those two RNAs base pair with each other. There's a lot of different ways to do this. Either way, as long as we initiate RNAI, all existing messenger RNAs of the same sequence are destroyed, and we shut down the expression of that gene on demand. So now we'll just wrap up this lecture talking very briefly about long non-coding RNAs. Again, this material is not in the older edition of the text, only in the newer edition, but everything we've talked about thus far is in both editions. At the complete other end of the spectrum from microRNAs and RNA interference is the long non-coding RNAs, and we mean long. To be classified as a long non-coding RNA, the molecule must, of course, not code for protein and be at least 200 nucleotides in length. It's believed at this point that eukaryotic cells, make over 8,000 different types of these long non-coding RNA molecules, and we think that the purpose of all of them are for gene regulation, although we are not sure at this moment. This is pretty new stuff here. The best, most classic example of a long non-coding RNA, especially one that plays a role in regulation, comes from the gene Xist, X-I-S-T. This is an X chromosome encoded gene. The RNA transcribed from this gene is a ridiculous 17,000 bases long. The gene is expressed very early on in the development of a female fetus. The Xist gene is activated on one and only one X chromosome, and this super long RNA molecule comes from this one gene. Amazingly, again, this long RNA, this single molecule over 17,000 bases, wraps or coats the entire X chromosome that it was transcribed from. Here what we see are DAPI-stained chromosomes. DAPI is just a non-specific DNA stain encoding all of the, dyeing all of these chromosomes blue. In violet is the Xist RNA molecule itself. You can see that RNA has completely enveloped, completely wrapped, almost cocooned a single X chromosome, whereas all of the other chromosomes are completely free of the Xist RNA. It is believed that by wrapping this X chromosome, the Xist RNA attracts enzymes to the X chromosome, and specifically these enzymes that are attracted are chromatin remodelers and other types of enzymes that compact the X chromosome into very, very dense, very inaccessible heterochromatin. Pull from some of your genetics background and see if you can figure out where this is going yet. This is the mechanism of X inactivation. We talk about X inactivation and dosage compensation in genetics, but this is how it happens. We're talking now about Mary Lyon's work on bar bodies, these fully compacted, inaccessible X chromosomes that occur only in female cells so that the dosage of X chromosome genes match in male XY cells and female XX cells. Again, females are XX. One and only one arbitrarily picked X chromosome expresses its Xist gene. That Xist long RNA is made and it coats that chromosome. That coded chromosome that expressed the Xist gene is compacted into a bar body. And so that cell now contains the uncompacted chromosome, X chromosome, that did not express the Xist gene in the fully compacted bar body. Again, it's arbitrary. So in another cell from the same individual, the purple X chromosome could express the Xist gene, itself be wrapped with the Xist RNA, and itself be compacted into a bar body. But the end result is the same, dosage compensation, one active functional X chromosome. It is thought that other long non-coding RNAs may silence other genes and other chromosomal regions in similar ways, but the truth of the matter is we simply don't know yet at this time. Please also keep in mind that not long ago, the DNA region that encodes the Xist gene would have been referred to as junk DNA. It doesn't code for protein. It had no known function. Yet we now know that this region has a purpose. It is crucial for X inactivation and dosage compensation. So always be mindful of the fact that junk DNA is not necessarily junk. It's just DNA with an as yet undefined purpose. So what did we talk about in this lecture? We started off talking about combinatorial control, where genes in eukaryotic cells are turned on or turned off by committees of proteins working together and individually voting on the status of a gene. Very few of our genes are regulated by a single factor or a single environmental stimulus. Instead, many proteins, dozens of proteins, come together to control the gene in a combination of their signals. That led us to the Mediator Complex, which is the integrator of these signals. Mediator is a huge protein complex that sits on the end of RNA polymerase II. It hears the signals from all of these different activators and repressors, and it relays that summative signal onto RNA polymerase II, allowing transcription to occur, or repressing transcription from initiating, depending on which way those scales have tipped. We use the fermentation genes, or hypoxic genes from yeast, as our example for coordinated gene regulation. This was the ROX1 TUP1 story. Then we moved on to talk about multicellular organisms, where the same basic rules applied as we had seen in coordinated regulation, but this time sets of proteins already made by a cell and a final single protein made could be the last determinative piece of the puzzle that seals a cell fate. We use the root of a UPS truck as our example there. We then leaped on to epigenetics and talked about how cells can have a regulation memory where they inherit the pattern of gene regulation from previous cells. This can happen through the histone code, which we talked about in previous lectures, positive feedback loops, which we defined very briefly, and DNA methylation, which we talked about in this lecture at a little bit more length. And then finally we wrapped up with post-transcriptional control, our UTO control, where cells have already allowed transcription to occur, but decide after transcription that they do not want to make that protein, and so they regulate genes after or post-transcription. We talked most about RNA interference, which is a very potent response for regulating gene expression at all, especially post-transcriptionally. We talked about the details of RNAI quite extensively. RNAI requires the formation of double-stranded RNA. Dicer processes this RNA. RISC uses this RNA to get the scent of other messenger RNAs that match its sequence and destroy them. And we even talked about amplification through RDRP. We wrapped up the lecture talking about long non-coding RNAs. We used Exist as our example, but there are other long non-coding RNAs that seem to do a similar function as well. This is a more newer material on the cutting edge of the field, and just an astounding and amazing example for how DNA of previously unknown function can have very severe and dramatic consequences in our cells once we understand what that DNA and the RNA that comes from it does. So that wraps up Chapter 8. We'll be moving right on to some of the experimental techniques that we can use with genes in our next lecture. That'll be parts from Chapter 10, Analyzing Genes and Genomes. Thanks for watching, and I'll see you all again soon.